This is my first video on the RNA processing process. In this first strand here, we see a 5' end and the poly A tail. The 5' end is made of guanine, and the poly A tail is made up of a repeating sequence of adenine bases. This is used on the RNA strand, so when it's transcribed, it protects important parts of genes that code for proteins, so that way they don't get cut off. So this repeating sequences of A's act as a buffer to protect the proteins in here. The UTR acts as the untranscribed region. We see some start and stop codons. And then in the middle here, we have the important region that includes protein coding segments. This is the most important part of this whole strand up on the top. And all this kind of acts as a buffer to protect the important strand. The second strand here we see is kind of similar. It starts off with a 5' end and it's ended with a poly A tail as well. These exons code for proteins while the introns do not. Introns are cut out because they really kind of don't code. So in this final strand, we see the 5' end, the poly A tail with just the exons that are left. We also have the 5' and the poly A tail to kind of act as caps.